Hello and welcome to our science segment. Today, we're diving into the microscopic world of Mycobacterium tuberculosis, or MTB, the culprit behind tuberculosis, a disease that continues to challenge global health. A key to MTB's success as a pathogen lies in its remarkable cell wall, a complex, waxy shield that not only protects it, but also interacts cunningly with our immune system. This wall is rich in unique lipids and sugars, which are now becoming prime targets for new diagnostic tools. Sugars, or glycans, are major players in the MTB cell wall. Think of them as the bacteria's handshake. They help MTB enter our cells and even manipulate our immune responses, sometimes blocking them altogether. Understanding these sugars is vital if we want to find better ways to fight TB. For a long time, studying these sugars was tough. They're not like DNA or proteins. They don't have easy-to-spot sequences, and you can't just tell the bacteria to make fluorescent versions of them. This made it hard to see what these sugars were doing during an infection. But there's exciting news. Researchers at MIT have figured out a clever chemical trick. They've developed a way to label a specific, rare sugar called MTX, which is found in a key MTB glycan known as mannose-capped lipoarabenomannan, or MANLAM. By using a special organic molecule called an oxaziridine, which reacts with these specific sulfur-containing sugars, they can attach fluorescent tags to MANLAM. This means scientists can now watch where MANLAM is in the bacterial cell wall and see how it behaves when MTB first meets our immune cells. What's really neat is that these sulfur-containing sugars are mainly found in MTB, making this technique highly selective. When they tried it on a harmless relative of MTB that lacks this MTX sugar, no fluorescent signal was observed. So what does this mean for diagnosing TB? Quite a lot. First, cell wall components like MANLAM are shed by the bacteria and can end up in patient samples, like urine. If we can specifically detect these MTB-associated sugars, we have new biomarkers for diagnostic tests. Current TB tests like sputum culture can be slow, and others like X-rays and molecular diagnostics aren't always available where TB is most common. A test that spots these sugars, perhaps in urine, could be faster, cheaper, and less invasive. There is an existing diagnostic in development that uses antibodies to detect MANLAM in urine. However, it tends to work best in patients with very active TB, particularly those who are immunosuppressed. The new small molecule sensor approach from MIT researchers holds the promise of a more sensitive test, capable of detecting MANLAM even at low concentrations. This could be particularly valuable for diagnosing latent or early-stage infections. Beyond just detection, the ability to track these glycans can also tell us how MTB's cell wall responds to antibiotics or when our immune system fights back. It could reveal how bacteria remodel their glycan barriers under stress, or how immune cells interact with these sugars. Some research even suggests that different mycobacterial proteins linked to the cell wall might be typical of active versus latent TB, offering hope for diagnostics that can distinguish these states. The unique sugars of the MTB cell wall are a hot area of research. These new chemical tools for labeling and studying them are paving the way for a new generation of TB diagnostics, ones that are rapid, sensitive, specific, and accessible, especially in regions hit hardest by TB. This work could significantly improve our ability to detect, treat, and finally, control tuberculosis. That's all for today. Thanks for listening.